Okay, so in the last class, we were looking at the structure of the chloroplast. So again, I'll just quickly revise that structure and then we will move on to the actual photosynthetic reactions, which is divided into light and dark. So in terms of chloroplast structure, if you remember, there was this outer membrane, inner membrane, then intermembrane space in between these two membranes. The whole of the inner core is called as stroma. Within, and this stroma actually contain different proteins, enzymes, and water and nutrients and so on. Within stroma, it had this disc-like structure. Individual disc is called as thylakoid, and a stack of disc was called as grana, and or grana if they are plural. And within the thylakoid, individually, there was this lumen again, which is a mixture of proteins, enzymes, and so on, including your different molecules of pigments, chlorophyll or whatsoever pigment. And the structure which is connecting two thylakoids is stroma lamellae, which is nothing but a tube-like structure. So these were the general structures of this chloroplast within a plant cell. So now moving forward with this particular part. So there are two phases of photosynthetic reaction, what we generally call as light photosynthetic reaction and a dark photosynthetic reaction. So we'll see them in different steps. So the thylakoid membrane, it actually houses the chlorophyll. So thylakoid membrane, in the sense, this green, dark green membrane, which is present in this disc-like structure, that is thylakoid membrane, and it contains this chlorophyll pigment and different protein complexes. Now, which will be these protein complexes? Obviously, the different enzymes or different proteins which are required for the photosynthesis function to happen. So they will be present, including photosystem 1, photosystem 2, and ATP synthase. That is, ATP stand for adenosine triphosphate synthase. So photosystem 1 is a particular protein complex which will absorb at certain wavelength of light, and same is the case with photosystem 2. It just absorbs at some different wavelength from the UV visible spectrum. So that is the identity, which are the proteins which are part of this, that we need not go into that detail, but just for your information, photosystem 1 and 2, these are different protein complexes which absorbs at different wavelengths. So that is what is photosystem 1 and 2. And then there is something called as ATP synthase. Now synthase is the common name for enzyme which will synthesize. Now what this synthase is specific for is ATP synthase. That means it will be responsible for synthesis of ATP, that is adenosine triphosphate. So these are the different protein complexes which are present in the thylakoid membrane, and which are specialized for the light dependent photosynthesis. So all these chlorophyll pigment as well as different protein complexes, they are specialized for light dependent photosynthesis. So light dependent means it is that photosynthetic reaction is dependent on the solar light or light energy, in short. So now what exactly happens there? So when a sunlight strikes the thylakoid, so directly it doesn't strike the thylakoid. Obviously, sunlight will pass through the atmosphere. It will fall onto the leaf structure. It will cross the epidermis. It will hit the chloroplast, and within chloroplast there is thylakoid. So when the sunlight strikes the thylakoid, the light energy which is present in the sunlight will excite the chlorophyll pigment. Now again, here I am assuming that you would know what is excitation and what is, I guess, what is the opposite to excitation? No, not de-excitation. But yeah, so I don't remember the word. In chemistry, this I'm talking about. So excitation refers to when electrons of a particular atom, when they gain a little more energy, they do not spin in a particular orbit. They go to the higher orbit, and from there they can even fall off the atom. Fall off the atom, that means they are escaped from that orbit and they are released. So that is the excitation of this electron. So in this case, when the light energy excites the chlorophyll pigment, which is again a compound molecule. So again, I will just show you the chlorophyll pigment, structure. Right. So if you look at this structure now, it's too complicated. But what I want to show here is nothing but that this one molecule is chlorophyll pigment, and you can see chlorophyll A and B. They have a subtle difference in terms of the one of the molecule being present at the R. I can't see where is the R. Where is it? I literally can't see. Maybe it's hidden behind this on top. Yeah, you can see it here actually. This is the R which is hidden there, right? So this is the general structure of chlorophyll molecule. So when the energy will fall down because of the too many double bonds and because of the electronegativity and electrophilicity of a particular atom, obviously there will be exchange of electrons between different atoms within the chlorophyll molecule and causing them to give up electron. So give up electron in the sense, like I explained that whole process, that energy will be taken from the solar light or the sunlight, that will be transferred to the molecule. That energy will be utilized by this molecule to cause a shift of the electrons within a certain molecule, or sorry, certain atoms within the molecule, and from there they can easily escape the electrons from the orbit. So that is what the second point talks about. Now the electrons whichever are released or whichever are given up by the particular atom from the chlorophyll pigments, they enter the electron transport chain. Now the electron transport chain, I assume that you may not have heard this word before. Okay? No. Electron transport chain is a series of chain reaction that happens within mitochondria. Electron, I don't know, I missed this spelling, electron transport chain. So this is the kind of reaction. Better picture, yeah, this is the kind of reaction, correct. So in this, what happens is whatever electrons are released, this whole complex of reaction is called as electron transport chain. Now why am I showing you this? Because these electrons, they will be utilized by this. You can see there is 1 and 3 and so on. So these are different photosystems. So these are the again another protein complexes in the thylakoid membrane. So these electrons will be taken up by this photosystem. They will cause the transfer of the energy from electron to different atoms. Finally, it will be utilized for breakdown of the H2O molecule, and that H2O molecule will be utilized for the synthesis of glucose. So that is in short a brief whole of this mechanism. So this electron transport chain that happens within the thylakoid membranes, within the mitochondrial membranes, and that's where these electrons go from once they are released by chlorophyll pigment. So I hope this part is clear. If at all you have questions, please do stop and ask me. So which is this ETC is a series of reaction that ultimately drives the phosphorylation of ADP, that is adenosine diphosphate. Phosphorylation means simple addition of phosphate group. So I will write down these steps on the board so that you will have more clarity. So first step, sunlight strikes on the thylakoid, because of which the solar energy will be like going to the chlorophyll pigments. That chlorophyll pigment will release the electrons. After release of these electrons, these will be trapped inside the electron transport chain, and literally the name says it, electron transport chain. That means they will transfer the electrons from one molecule to other molecule. That is what is electron transport chain. So now what exactly happens in that? You know, any bond formation within a biochemical molecule will need certain energy. So this phosphorylation, which is addition of phosphate to the ADP, is an addition reaction. Addition reaction in this is some molecule is getting added to the some other molecule. So that way ADP, adenosine diphosphate, becomes adenosine triphosphate, that is ATP, which you commonly know as the energy molecule of a cell. So that is what is phosphorylation reaction, that is addition of phosphate group to the ADP molecule. So that is the stage. Now the electron transport chain also results in the production of reducing agent. Because it carries the reduction reaction within the particular cell, and that is known as NADPH, which means nicotinamide adenine dinucleotide phosphate. You will have to remember all these terminologies because they are important in the context to the syllabus. So that is NADPH, sorry, not NADH, but NADPH. So that is nicotinamide adenine dinucleotide phosphate. And this H actually stands for hydrogen, which is already there with this nucleotide molecule. So you don't specifically mention it in this name. And that is why it remains nicotinamide adenine dinucleotide phosphate. So there is another molecule which is NADP plus or NADP in short, which also has the same name, but this is the reduced form of that NADP. So reduced form again, that means it has gained certain electron from some other molecule, and that's why it is reduced form. So that way the whole chain of reaction happens in the light photosynthetic reaction. Any questions so far? Anyone? This can be a little what do we say, a boring point, but I hope you all are listening and trying to understand this whole concept. Later on it will be very difficult to understand if at all you try to break your head on it. So if you have any questions, please feel free to ask. Okay, so now so in this photosynthesis, it actually begins with the light reaction. Light reaction, that means the whole of the photosynthesis process, like I said, is divided into light and dark. So one cycle of photosynthesis, the first step is the light reaction, and which is carried out only during the day. An obvious reason, because of the presence of sunlight during the day. So in plants, the light dependent this photosynthetic reaction takes place in the thylakoid membranes of the chloroplast. Again, one of the differentiating factor from dark photosynthetic to light photosynthetic, which we'll see later. But dark photosynthetic reaction happens in the thylakoid membrane, right? Why does it happen in the thylakoid membrane? Because obviously the chlorophyll pigment is present there. Why only during the day? So the reason lies in during the day there is sunlight. Why at thylakoid? Because there is a presence of chlorophyll pigment and so on. Then, so the grana, which is membrane bound sacs, which is, or rather the stacks of thylakoid, which are the structures present within the thylakoid function, they act by gathering the light, and that particular protein complex or that particular molecular complex is called as photosystems. So it's not just one chlorophyll molecule or one thylakoid disc-like structure present in the leaf or present in the chloroplast, because there are almost quite a few thousands of individual. So there will be thousands or even more than 1,000 of chlorophyll pigment. And same way there are too many thylakoid structures within most of the cells of the leaf. So they all will generate electrons. All these electrons, whichever are generated within this thylakoid by the chlorophyll, they will be gathered in a photosystem. Again, photosystem is nothing but it's a protein complexes, which is helping to gather all the electrons or gather all the light or gather all the energy coming to them. These photosystems, they have large complexes of pigment and protein molecules present within the plant cells, and which play a primary role during the process of light reactions of photosynthesis. So that the main function of photosynthesis is to gather the light and keep it kind of reserved with the photosystem protein complexes. So that is the first stage. Now, in general, what we know of, there are two types of photosystem 1 and photosystem 2. Again, the difference I will show you here in the diagram. So, photosystem 1 and 2 structure. So, no, not these ones. I guess these ones. Yes. So, if you can just have a look at without getting overwhelming. So, you can see there are too many proteins involved in photosystem 1. Right? Sorry, this is photosystem 2. This is 1. But either ways, there are many proteins which are shown here like a disc-like structure. They are involved in these two, photosystem 1 and photosystem 2. Here also you can see there are quite a few proteins which are like labeled as photosystem 1 and 2. So what I want to say is it's not just a complex of a single or two protein. It is a complex of many proteins. That is what we call as photosystem 1 and 2. Now these photosystem, these are the system which are absorbing the light. So under the light dependent reaction, that means during the day, and that is when the light photosynthetic reaction happens, which is also called as light dependent reaction. The light energy is converted to ATP and NADPH. How it is converted into ATP, that is what we saw in the earlier slide. So that the light energy which is trapped in the electron transport chain, that will be transferred to the photosystem 1 and 2, which will help in the phosphate addition to ADP molecule, creating ATP and NADP. So these are the two energy molecules. Commonly what we know is ATP is the only energy molecule. That is the common consensus. But within our body, there are three main energy molecules. One that you know of commonly is ATP. Second one is NADPH. And the third one is called as flavin adenine mononucleotide molecule. It's called FAM or FAH and so on. So there are again different types of it, subtypes of it, I would say. So there are three different energy molecules, out of which two molecules, that is ATP and NADPH, they are synthesized or they are produced in the light dependent reaction or light photosynthetic reaction. And these are used, these molecules, whichever are generated, they are used for the second phase of photosynthesis, that is dark photosynthetic reaction. So during the light reaction, ATP and NADPH, they are generated by the electron transport chain, and water is used and oxygen is produced. Now the reaction looks like this. So two molecules of water, two molecules of NADPH, which is a non-reduced form of energy molecule, and it uses 3 molecules of ADP plus 4 molecules of phosphate. The ADP and phosphate will add, it will form 3 ATP. H2O, the H will go to NADP, and it will form NADPH. So that way, and then 3 molecules, and the oxygen is released from this reaction. So that is the overall light photosynthetic reaction which happens in the plant. Again, any question here? Anyone? FA. So let me show you that as well, because ATP NADPH and FADH2 or FADH2. Let me put it that. Okay, I don't see the structures as such. Let me put it. It's called flavin adenine dinucleotide molecule. So yeah, you can see here flavin adenine dinucleotide, and the structure is something like this, which is again, the structure is not important for you people, but yeah, you need to know that there is something called as FADH2 as well, apart from NADPH. Yeah, again, this is the structure of NADPH. So there is like this nucleotide base which is present in your DNA or RNA, which is adenine here in this case. There will be a sugar molecule. You can see there are two sugar molecules, one here and the other one here, and then there is a phosphate, and then there is another molecule attached to it. So depending on what molecule we are talking about. So this is the general structure of NADPH. So that is dinucleotide. Right, so these are the different energy molecules. Anyone else? Any other question?
Sir, do we need to memorize that equation?
Yes, this. So the equation will be asked in terms of what is the difference between light photosynthetic reaction equation with compared to the dark photosynthetic reaction. So then this will be important. Or what is the difference between the products of light photosynthetic reaction and dark photosynthetic reaction? So ultimately you will have to write this equation and identify and understand it that way. But simplest thing what you need to remember as of now here is, we know photosynthesis at the end forms glucose, right? Half of that step is generation of energy and generation of oxygen. And that reaction is the first part of the reaction, that is light photosynthetic reaction. So that is when a balanced equation you need to know. It's not just the equation. So you cannot write H2O plus NADPH plus ADP. You have to write these prefixes in terms of numbers as well, so that the reaction is balanced. Okay, why it's not going forward? Okay, yeah. So this is the diagram which is kind of showing the photosynthetic reaction. So what they are trying to show here is one of the thylakoid, right? And the membrane of that thylakoid is being enlarged in this big box. Yeah. So within this big box, you can see the membrane, right? That is the thylakoid membrane. Within this thylakoid membrane, this is the stroma, that means the outside the thylakoid, and this yellow part is inside. You can see yellow here as well, which is the lumen. So this yellow part is the lumen of the one thylakoid structure. So that is the inner thylakoid space, or we can simply call it lumen, and outside the membrane there is a stroma. So this ETC, electron transport chain, is present right in the thylakoid membrane itself. So what will happen again? Thylakoid membrane will, this green part you can see, it will be striked with the light energy. It will release electron from the chlorophyll. They will be transferred to the electron transport chain, and that electron transport chain will cause ultimately the generation of ATP, which you can see here. Which is with the help of enzyme, which is, we call it ATP synthase. Here it is called as ATPase complex and so on, but that doesn't matter. You can call it ATP synthase. And then this energy whichever is utilized, that will be used for the second photosynthetic reaction, that is dark photosynthetic reaction. So the enzyme will also catalyze the reaction where ADP and inorganic phosphate will produce the ATP. So that is again overall overview of the light photosynthetic reaction. Any question here? So there was some reference. Yeah, I don't know what is this reference. I don't remember. Oh, no, it is not taking. It has to be like this. So this reference is from ICSE solution. Oh yeah. So this one is one of the good explanation that has been given regarding light photosynthetic reaction. So again, if at all you are interested in reading more about this, you can look at this particular reference which is cited here. Then we will move forward to dark photosynthetic reaction. Now this is one of the most important part of photosynthesis, whole of the photosynthesis. So whatever ATP and NADPH which are synthesized in the light photosynthetic reaction, they are used in the dark reaction, which is also called as light independent. Independent means ultimately dark reaction. Light photosynthetic reaction is also called as light dependent. This one, that is dark photosynthetic reaction, is also called as light independent, which is very obvious, in which carbon dioxide and water, they are assimilated into organic compound. Assimilated means they are put down together in like or what you say, we make something or create something. So from carbon dioxide and water, they will form a certain structure of organic compounds. So that is what is the dark photosynthetic reaction. And like I said, it is the second half of the photosynthesis process where the glucose is produced. So the light independent reactions of photosynthesis, they are carried out in the chloroplast stroma. The most important difference, like I mentioned, the light photosynthetic reaction happens in the thylakoid membrane, while the dark photosynthetic reaction happens in the stroma, that is within the chloroplast, outside the thylakoid membrane. So if I show you this diagram again, this one where the yellow part is, or rather where the green membrane is, that's where the light photosynthetic reaction will occur. And in the second part, which is stroma, that is outside this thylakoid structure. So outside this grana, there is some space. Within that space, the dark photosynthetic reactions will occur. Now, this stroma contains different enzymes, and one of them is called as ribulose, which is a sugar 1,5, which is indicating the position of this ribose sugar or ribulose sugar. Bisphosphate, that means two molecules of phosphate, they are attached to the first carbon and fifth carbon. Carboxylase, that means this reaction is breakdown of the carboxyl group. So that carboxylase, ASE, that indicates sometimes the breakdown or removal. So carboxyl group is removed, or oxygenase, that means oxygen is removed. So that this name Rubisco 1,5 bisphosphate carboxylase or oxygenase, it is short form as Rubisco. That is the name of the enzyme which catalyzes the dark photosynthetic reaction in the stroma of the chloroplast. Now what does exactly it do? So Rubisco catalyzes the first step of carbon fixation. Carbon fixation, what we call as the environmental carbon dioxide, which is taken by the leaves, and that is being produced into glucose, which is a carbon fixation process. That means carbon was randomly moving around in the atmosphere, that is being fixed at one place in the organic compound matter within the leaves. So that's what is carbon fixation. And this is a part of Kelvin cycle, or which is also called as Kelvin and Benson cycle. because these were the two guys who had invented this. So this Rubisco will catalyze the first step of the carbon fixation. I guess, have I? Yeah, I have it here. But let me also show you a little complicated version of it, just for the sake of understanding that whole of the purpose. I'm not too sure where exactly is this. So just give me a moment. Is Rubisco here? Not really. Ideally, it should be shown here. Yeah, here it should be. Yeah, you can see here. So the atmospheric carbon is added to a particular uh, uh, RUBP, which is like kind of 1, 2, 3, 4, 5. This is a ribulose sugar, which is a pentose sugar. Pentose means 5 carbon sugar, right? So when a 5 carbon sugar, which is a byproduct of this Kelvin cycle, it is added with carbon dioxide, which has 1 additional carbon. What you get is 6 carbon sugar, right? That is nothing but your glucose ultimately. So this glucose, that is the first reaction of this addition of carbon dioxide to the pentose sugar molecule, which is ribulose sugar here, ribulose bisphosphate. So that reaction is catalyzed by Rubisco enzyme. Okay, so this particular reaction, the which says Rubisco catalyzes the first step of carbon fixation in the Kelvin cycle. So that means the Kelvin cycle, which is the primary pathway of the carbon transport in plant. Carbon transport, again, it goes back to the same fact that it is a carbon fixation process. From the atmosphere, 1 molecule of carbon dioxide is put down in the pentose sugar, that becomes 6 carbon sugar, and that is then converted into the glucose molecule. And that glucose is again utilized for energy generation. And then this goes on. So because it is a cycle or cyclic process. So among the so-called C4 plants, now again this is a little complicated part, you need to pay more attention. There are 2 types of plants in general, depending on which mechanism they undergo, which is called as C4 and C3 plants. C3 and C4 plants. So these are the 2 different types of plant in which specifically, where is it shown here? I don't think so. It is given clearly here. Maybe here this should be good. Glucose, C3, C4. Yeah. Just a subtle difference. Open image in new tab. What happens is within the C3 plant, again the same ribose sugar, which is a pentose sugar, will get 1 molecule of carbon dioxide. It will form 6-membered compound, which is glucose. That is C3 plants, which is normal Kelvin cycle. In C4 plants, what happens is, because they don't have sufficient enzymes, what they do is they create an intermediate, which is a 4 carbon. So here the 3 carbon molecule is formed and 3 plus 3 becomes 6. Here a 4 carbon intermediate is formed, and with that addition of carbon dioxide molecule, it forms 6 glucose molecule. Now I am telling you in a very simplistic way. Otherwise, and then there is of course the third one, which CAM plant, which we are not talking at all about. So there is the C3, which means 3 carbon intermediate. C4 means 4 carbon intermediate is formed. Ultimate product and the starting reaction product, starting reactants, they are the same. Just the intermediates are different. That's why they are called as C3 and C4 plant. So the statement goes like, so in the C4 plants, the initial carbon fixation step and the Kelvin cycle, they are separated spatially. Now, I assume you would know the meaning of this word. Spatially separated means they happen at different places. That is what is difference of place or different of space. So that spatial difference happens at different places, And what are those two reactions which happens at two different places? That is the first step of the initial carbon fixation, that is where the oxygen gets added, and the actual Kelvin cycle, that is the rest of the Kelvin cycle where it happens. So these two happens at two separate places. So the carbon fixation occurs via phosphoenol pyruvate, which is PEP, carboxylation. So carboxylation is addition of carboxyl group, which is COO group, and that COO is coming from your carbon dioxide. So phosphoenol pyruvate. I don't know if I will show you the structure, it will become a little easier. I feel it will just get more complicated, so that's why I'm refusing to show you the structures. What is this? Non-cyclic, we don't want to go there. Let me show you the PEP structure in any which case. If at all those who get it, we will see. You open. So PEP, no, carboxylation. So not here, not this one also. I hope I'll find a easier one. Yeah, okay. So open image in Newton. So you can see this is a simpler molecule, right? This C double bond O minus, this is one carboxyl group, and then C double bond O, that is another. So this is pyruvate molecule, right? And because there is a phosphate group and this double bond keep transitioning between, so that is why it is called as phosphoenol. Enol, that is the alcohol type, that is hydroxyl type and keto type. But again, that is too much of chemistry to understand. What you need to know is this is a phosphoenol molecule. When the carbon dioxide gets added to it in with the presence of water, of course, it becomes like this oxaloacetate. And this is like, if you see 1, 2, 3, 4 carbon molecule, right? So that is what is the C4 plant happening. And again, this phosphate is released. And this phosphate which is released is again utilized for the formation of ADP to ATP. So adenosine diphosphate, it needs one additional phosphate to become triphosphate. So that is where this will get added on. So this is the particular chemical reaction where the oxaloacetate, which is a C4 compound. C4 means carbon, 4 carbons are there. So that's where this carboxylation reaction occur. So the carbon fixation occurs via phosphoenol pyruvate carboxylation in the chloroplast, which is located in the mesophyll area of the leaf structure. While malate, which is again another compound, a 4 carbon product of that process, is transported to the chloroplast. So like I said, this reaction has 4 carbon compound. So malate formation in photosynthesis. So malate is another type of compound which gets formed. Yeah, this is even better diagram. So let me use this one now. So what they are talking about is, this is the overall C4 pathway. This is the first step of the Kelvin cycle in C4 plants, right? So what happens is PEP, phosphoenol pyruvate, which is 3 carbon compound. After addition of carbon dioxide, it becomes 4 carbon compound. And this reaction is catalyzed by PEP, phosphoenol pyruvate carboxylase, right? So after this 4 carbon compound, it gets converted into malate. So that is one type of reaction, which is just a simpler, I guess, addition of hydrogen to oxaloacetate, and then it forms malate. So this malate gets transferred to the cytoplasm, right? Where is it? So where was that? The 4 carbon product of that process is transported, sorry, not cytoplasm, the chloroplast in the bundle sheath cells, right? So this, all these things, they were happening in the stroma of the chloroplast cell. So that is where oxaloacetate to malate formation happens. Malate will cross the membrane, and the reason why oxaloacetate, even being 4 compound, it does not get transported is because of its chemical nature. So obviously the hydrophilicity, hydrophobicity, all these factors, they need to be considered. So in short, what we need to know is, in C4 plants, there is formation of the C4 intermediate, which is oxaloacetate and malate. And because oxaloacetate cannot get transported across the membrane, it forms malate, and malate gets transported across the membrane, which ultimately enters your what you say, Kelvin cycle, and then it continues with the glucose formation. So that is the general reaction. Again, I can totally understand if you are not getting all of these reactions at one go, but all of this we have to know in this dark photosynthetic reaction. So again, let me revise this part quickly. So light photosynthetic reaction, we know the sunlight will fall onto the thylakoid membrane. Ultimately, it will fall onto the chlorophyll pigment. It will release electrons. These electrons which are released will be utilized in the formation of ATP and NADPH, right? And oxygen is released in this. Now, whatever this energy is generated here, that is ATP and NADPH, that will be utilized for the next reaction. So next reaction, so actually if you look here, in this case, where is the ATP? Yeah, you can see ATP is getting utilized, and then it is again forming AMP, which is adenosine monophosphate. So there are 3 forms: adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, 3 forms. So ATP is getting utilized in this second part of the reaction. So that is what is being said there. So after the formation of energy, it will enter into the dark photosynthetic phase of the photosynthesis. Now in this, the first step is catalyzed by Rubisco, which is addition of carbon or carbon fixation into the Kelvin cycle. Now in that, there can be 2 different pathways. One can be C3 and the other can be C4. In C3, there are 3 carbon compounds which are formed. In C4, there are 4 carbon compounds which are formed. Specifically, this carbon fixation occurs via this PEP carboxylase enzyme in the chloroplast, and that will add carbon dioxide. Then oxaloacetate is formed, and from oxaloacetate, what we get is malate, which is the another 4 carbon product. And then that is transported to the chloroplast in the bundle sheath cells, which is a type of cell, which is a type of cell within the leaf structure, and that is where the Kelvin cycle is carried out further. That means the glucose formation will be carried out further. Any doubt here, anyone so far? So we are yet to see the final reaction. If anyone has any question, you can ask. Otherwise, we will stop here for today, because it will be just too much. Yeah, it is too much. Actually, it is too much. I would disagree on that part. Okay.